So, hello everyone. Let's continue with the lysosomes. We have previously discussed that the lysosomes are involved in degradation of lots of different products. They have got acid hydrolases inside them, and these acid hydrolases can destroy proteins, nucleic acids, and lipids. There's a wide variety of these enzymes found inside the lysosomes. Um, so in addition to performing these functions of degradation, lysosomes are also involved in lots of different processes. They're involved in performing different functions, and this is especially carried out by the lysosomal membrane proteins. What we have previously discussed that they're involved in phagocytosis, because when a cell engulfs some solid particle, it wraps it in order to form a phagosome, where the plasma membrane encircles around this engulfed particle. This phagosome is converted into phagolysosome by the fusion of lysosomes with phagosomes. So that means the lysosomes fuse with the phagosomes, pour their acidic enzymes into the phagosomes, and this phagosome is converted into phagolysosome because this is a phagosome which contains content from the lysosome, means it contains these acid hydrolases which can now destroy this engulfed particle. Lysosomes are also involved in macroautophagy, where an isolation membrane, generally smooth endoplasmic reticulum, it wraps around the old components of the cells and leads to the formation of what we call autophagosome. This autophagosome is converted into autolysosome by the fusion of lysosomes with autophagosomes, and now this autolysosome can degrade those encircled or wrapped compounds into small particles. So proteins, lipids, and nucleic acids can degrade it inside these vesicles. And then we have associated with macroautophagy, we have this chaperone-mediated autophagy of cytosolic proteins, which are so basically the proteins which are synthesized by the free ribosomes in the cytoplasm. They are degraded via lysosomes, while the proteins which are the misfolded proteins produced in the rough endoplasmic reticulum, they are destroyed via proteasome degradation, what we have previously discussed. So all of these processes, phagocytosis, macroautophagy, or chaperone-mediated autophagy, all of them are basically mediated through LAMPs. LAMP stands for lysosome-associated membrane proteins. So basically these LAMP proteins help in fusion of the lysosomes with either autophagosomes or with the phagosomes. So LAMPs basically help in membrane fusion between the phagosomes or autophagosomes and the lysosomes. Then we have CLC7 proteins. These are transmembrane proteins, and these proteins help in acidification of the lysosomes. So basically they are like antiporters. Antiporter means they will kick one anion or cation out, and then they will put the other one inside the lysosomes. So in case of CLC7 here, chloride would be exported out of the lysosome into the cytoplasm, and protons from the cytoplasm would be poured into the lysosomes, and this would increase the acidification of the lysosomes. So basically they're antiporters and they're transmembrane proteins. They're channel proteins basically. So they move one thing in for the other one that moves out of these lysosomes. Lysosomes also play a role in exocytosis and plasma membrane repair. So they can produce exosomes which can fuse with the plasma membrane, and this helps the membrane in the repairing process. We have seen that endocytosis or phagocytosis leads to the reduction of plasma membrane, because if the cell keeps engulfing product from the outsides and keeps producing these vesicles, then with the passage of time this plasma membrane can shrink and the cell can become smaller. But this is again compensated by both by the endoplasmic reticulum, which keeps producing these vesicles which fuse with the plasma membrane, as well as by the lysosomes, which produce these exosomes, and these fuse with the plasma membrane, and this helps in the repair process of the plasma membrane. Synaptotagmin proteins are also involved in this. These are the proteins which are involved in lysosomal exocytosis and plasma membrane repair. Synaptotagmins are also involved in neurotransmitter release near the synaptic junctions. The other function that the lysosomes play is in the lysosomal cell death. Lysosomes contain certain proteases, what we call cathepsins. Basically, these proteases, they work at an acidic pH, and generally they're not destructive to the cells. But release of lots of cathepsins in response to initiation of the cell death pathway can lead to the destruction of the cells. Not all the cathepsins are, so most of the cathepsins, they reside inside the lysosomes and they work at lower pH, but some cathepsins like cathepsin K can also be found in the extracellular space. So anyhow, these are proteases basically, and they perform their function in destruction of various types of proteins. So there are more than a dozen cathepsins, different types of cathepsins, which perform different functions. They have their own substrates. So they do play a part in lysosomal cell death. And this is the reason they are also known as suicide bags. Another major function that the lysosomes perform is cholesterol homeostasis. So basically, this cholesterol homeostasis is mediated by Niemann-Pick-C1 and Niemann-Pick-C2, these two proteins. Let's look at this thing on the next slide. The cholesterol basically keeps moving inside our blood in the form of LDLs, what we call low density lipids. So basically these are lipoproteins, LDLs. What do lipoproteins do? Lipoproteins carry around cholesterol or lipids to different parts of the body, as well as into the cells. So cholesterol basically remains associated with these proteins and with fatty acids. So what we call LDLs, with these proteins. So these low density lipids, or the lipoproteins which are carrying lipids, they're of different types. They could be very low density lipids, what we call VLDLs, and then we have low density lipids, LDLs, and then we have high density lipids, HDLs. So LDLs are also known as bad cholesterols, because if they are not absorbed by the cells, this cholesterol stays inside the blood vessels, and it keeps building up into the blood vessels, into the walls of the vessels. So the blood vessels become squeezed, and they become narrow, and this may lead to different types of problems with your heart, and it can also raise your blood pressure because of the deposition of LDLs, or because of the deposition of the cholesterol in your blood vessels become squeezed. So generally, lysosomes play a major function in cholesterol homeostasis. So how do they do this? These LDLs, they are absorbed by the cell via receptor-mediated endocytosis, and this receptor-mediated endocytosis is achieved because of the presence of these proteins in LDLs. So proteins help these lipids enter into the cell via receptor-mediated endocytosis. So cholesterol is found in the form of these LDLs, or what we call cholesterol esters. We know that lysosomes contain acid hydrolases, and they also contain lipases, proteases, lipases, nucleases, etc. So acid lipases would remove fatty acids from the cholesterol molecules, and then this cholesterol becomes bound with a lysosomal soluble protein, what we call NPC2, Niemann-Pick C1, Niemann-Pick C2. So NPC2 binds with cholesterol and carries cholesterol to NPC1. So the difference between NPC1 and NPC2 is that NPC2 is a soluble protein and remains inside the lysosomes, while NPC1 is a transmembrane protein. It's a membrane-associated protein. And basically, binding of the cholesterol with NPC1 helps in shuttling or export of the cholesterol from the lysosomes to the ER membrane. So both NPC2 and NPC1 are basically, these are lysosomal proteins which perform their function in exocytosis, in efflux of the cholesterol from the lysosomes, and this cholesterol can then be utilized in the ER. So that's how lysosomes perform their function in cholesterol homeostasis. Another function that they perform is MHC class II dependent antigen presentation. So basically, in case of immunity, we have certain specialized cells which can engulf different types of microbes. So when these microbes are engulfed, basically these microbes are degraded into pretty small particles with the help of acid proteases, which are found inside the lysosomes. When a microbe is engulfed by the lysosomes, this microbe is engulfed in the form of a phagosome. And when a lysosome fuses with this phagosome and it forms a phagolysosomes, then proteases inside this vesicle, they start degrading these pathogens into smaller peptides. And these peptides are then associated with MHC class II molecules. And the exocytosis from these lysosomes then leads to the formation of these MHC class II compartments, where MHC class II receptors, they bind these smaller peptides to themselves, and during the exocytosis process, the MHC class II molecule, it releases the other degraded peptides, and some of them, the antigenic peptides, they remain associated with the MHC class II molecules. And these MHC class II molecules are then recognized by some other cells of the immune system. And this is how the immune system knows that this cell has got some, or there's some sort of a pathogen or foreign particle living inside the body. So these antigens are then presented to the cells of the to the effector cells of the immune system, which then perform their function to degrade these pathogens. Not this pathogen, but the identical pathogens which are living somewhere else inside the body. So this is how lysosomes perform their function in immune system. Then we have these VATPases. So VATPases are basically, these are the what we call these are the polar opposites of ATP synthase. We will study ATP synthase later. So basically, what do? They perform their function by hydrolysis of the ATP molecules, and what they do is that hydrolysis of ATP molecules basically leads to the influx of protons into the lysosomes, and this again lowers the pH of the lysosomes and makes this environment more acidic. So both so V stands for vacuolar type proton ATPase. So basically hydrolysis of ATP leads to the movement of protons from the lysosomes, from the cytoplasm into the lysosomes, and this then elevates the acidic environment of the lysosome, so it makes it more acidic, so that hydrolases can perform their function at their optimum pH. So these LIMPs, which is lysosomal integral membrane proteins, they perform their function in import of newly synthesized hydrolyses. So these hydrolyses, these enzymes these are synthesized either by the free ribosomes or by the ribosomes on the endoplasmic reticulum, and then they are transported into the lysosomes. So basically, these integral membrane proteins help the lysosomes take in these newly synthesized hydrolyses. So these are different functions performed by different lysosomal membrane proteins. So CD63, these are basically glycoproteins, which are known to complex with integrins, and these proteins mediate signal transduction events that play a role in the regulation of cell development, activation, growth, and mortality. So they perform different types of other functions. So we have learned today that the lysosomes they contain different types of membrane proteins which perform different functions, and these functions can be associated with phagocytosis, with macroautophagy, with chaperone-mediated autophagy, with lysosomal exocytosis, plasma repair, neurotransmitter release, lysosomal death, lysosomal cell death. They're involved in cholesterol homeostasis. They're involved in adaptive immunity by the release of amino acids and exocytosis, and they're involved in MHC class II dependent antigen presentation. So there are also several other functions that are performed by the lysosomes. So thank you very much. I think that's it for today's lecture. So if you have any questions and comments, do post them, and I would be glad to answer those questions. Thank you very much.